So my name's Ben. I just finished my PhD at Stanford in the genetics program in Omnibot's lab. And in that program, I studied the human microbiome. And I'm really interested in a few aspects of the microbiome that we'll talk about, but also how we study the microbiome through bioinformatics and genomics methods, and also how people have now started to commercialize the microbiome, both for medicines, for personalized health, personalized nutrition, all that. So I will talk about a few things today. First is what we know about the human microbiome, how we learned it, some of the basic facts that you may or may not have heard, how we study the bacteria, phages, and fungi in the gut. Then I'll talk about my research that I did in Omnibot's lab about humans acquire and how humans acquire and transmit members of the gut microbiome. Then I'll talk about some microbiome companies, both public and private, that I've seen and what I really find interesting in the space. And then we'll have plenty of time for Q&A afterwards if you have any other questions. So starting off with what we know about the human microbiome, collectively, the microbiome is all of the microorganisms that live on and inside the human body. And the greatest density of these microorganisms is in the human intestine. We call this the gut microbiome. So there's over 10,000 different species of bacteria, fungi, archaea, and viruses that can live in a single person's gut. And these microbes total roughly the same number of cells as in the human body, but they're generally much smaller, and they collectively weigh only 200 grams in the average person. But where the microbiome really stands out is the gene content. So humans only have about 22,000 genes in their genome that encode all the instructions for making cells, conducting metabolic processes, defining your reproductive pathways, all that. But collectively, the gut microbiome can have over 150 times the number of genes that are in the human genome. So there's this huge wealth of unexplored diversity, most of which is undiscovered, all residing within the gut in every person. For a closer look at the gut microbiome, I love this microscopy image out of the Sonnenberg lab here at Stanford. And this is a section of the mouse intestine where the host intestinal cells are labeled in blue on the bottom left. The mucus layer excreted by the intestinal cells is labeled in green. Debris from food is labeled in yellow, and bacterial cells are labeled in red. What I hope you can appreciate is how abundant and pervasive the bacteria are. They live on and inside the mucus layer, right next to the host intestinal cells. All of these bacterial cells living within a person's gut aren't just passive bystanders. And collectively, the gut microbiome has massive impacts on human health, including the ability to regulate the development and functioning of the immune system, extracting certain nutrients from food that the body can't process on its own, protecting the body from invasion with certain infectious organisms, affecting how the body responds to certain cancer therapies that reactivate the immune system to target tumors. This is one of the most interesting things I think about the gut microbiome. We'll talk a bit about that later. And finally, influencing the brain and the nervous system, including producing up to 95% of the body's serotonin is produced within the gut. And this is something really interesting that has only started to come out in the past 5 or 6 years called, you might have heard of it, called gut-brain axis. And there's new studies that suggest that the microbes that are living in the gut can produce molecules, some of which can cross the blood-brain barrier and may have impacts on certain neurodegenerative diseases like Parkinson's and also certain behavioral diseases and traits. But actually, there was some recent research published last week that showed that the link between the microbiome and autism may not actually be mediated by this kind of gut-brain axis metabolite production, but may be entirely mediated by different behaviors in autistic children that lead to different diets that they choose to have that lead to different gut microbes. So there's really this kind of like complex association, especially when we're talking about the gut-brain axis. And this is just a headline from 2015, which I think is one of the earliest headlines that I've been able to find, where scientists were first starting to suggest that the different bacteria in your gut can produce produce metabolites that might be actually functioning the way your mood and perhaps even the choices of food that you make. Where my research started to come in was about how the gut microbiome is dynamic and changes over time. So starting at the first days of a baby's life, the microbiome develops into an increasingly complex community. Most of the first microbes colonizing an infant are the result of direct transmission from their mother. And this direct transmission is especially important for colonization with the bacteria Bifidobacterium infantis, which has the unique ability to digest human milk oligosaccharides, which are nutrients in human milk that the body can't process on its own. And so where close contact between a mother and infant is responsible for healthy microbiome development, other exposures during infancy and childhood can have a negative effect. And for example, both C-section birth and formula feeding can hamper the development of healthy gut microbiome, although the change is not permanent. And antibiotic treatment and malnutrition early in life can also have detrimental effects. So once the human reaches adult or adolescence, though, most of the microbiome maturation is complete and the community is well established. It's understood that barring major modifications like broad spectrum antibiotics or fecal microbiota transplant, the microbiome of adults doesn't change much over time. So even drastic changes in diet, such as going from vegetarian to an omnivore or carnivorous diet, they may change the microbial community in the short term, but many diets have been shown to alter the microbial community over many years. And if we think about ways that we can modify the microbiome, so that's either going to be taking it away from a potentially diseased state into a healthy state, or changing something about the microbiome that may help you digest certain food better or ameliorate symptoms of irritable bowel syndrome. There's a few different ways that we can do that, and these three are, healthy ways that we can modify the microbiome. I didn't include in here antibiotics, which, or especially oral antibiotics, which people often take and can kind of kill off all the healthy colonizers. But the three main ways that we think about modifying the microbiome today is what's called a fecal microbiota transplant, or FMT. You've probably seen this in the news for treatment of Clostridium difficile, which is a antibiotic-resistant pathogen that lives inside the gut. And often, after multiple courses of antibiotics, all of your natural microbiome colonizers are killed off, and only this C. diff persists. So this is where a fecal transplant, where you take the entire microbial community of the donor and you put it into a recipient. This is one of the most effective ways to cure C. diff. And this FMT is interesting because it's the entire community that's passed from donor to recipient. And you often get good engraftment, meaning that the microbes take residence inside the host and they don't just kind of pass through. But you also don't really have control over which specific microbes are going to be given to the recipient or which are going to persist long term. And that means you have to be really careful about safety. You don't want to be giving people potentially more antibiotic resistant or harmful bacteria. Probiotics are often common either in a pill form or part of active culture yogurt or in fermented food. These can definitely shape the microbial community, but oftentimes many probiotic supplements that people take don't actually engraft long term. So while you may find evidence for the probiotic while you're taking it and it'll bloom and live in your gut for a short period of time, it won't persist long term after you stop taking the supplement. And finally, which is a new area of research, is called prebiotics. So it's the opposite of probiotics, where we're adding things before the bacteria, so the actual nutrients that the bacteria need to live. And so this is an active area of research of which there's not too much being that is well known or has been in clinical trials yet, but I think it's an interesting area where you could give a nutrient that certain bacteria like better and shape the community that way. So moving on to how we study the human microbiome. Most of what I do revolves around DNA sequencing and not the RNA, but the actual DNA genomes of the microbes in the samples that we have. So in our lab, we collect lots of stool samples or poop samples from patients in the hospital and healthy donors, healthy volunteers, et cetera. And each stool sample is taken as a collection of all of the microbial, the different microbial cells that are inside a person's gut microbiome. Sorry, Grace, can you mute yourself?
Is there a question? Okay, go ahead.
Okay, so stool sample is a collection of all of the microbes that are in a person's gut. You also have obviously a post DNA as well, but we're only going to be focusing on the microbial side. So each bacterial genome in the stool sample that we collected has a DNA genome, and that's typically a circle, somewhere around a few million base pairs in length. So the DNA is made up of the four bases, A, T, C, and G. So we extract that DNA into solution from the stool sample. We shear it up into small pieces, and then we read out the letters on a DNA sequencing machine, typically made by Illumina. We're also using some newer technology by Oxford Nanopore, which is a company based in London that has the ability to read much longer fragments of DNA. And so from this experiment, we typically get several million sequencing reads. Each is roughly 150 letters in length. And these describe the genomes of the microbes in the sample, and therefore the microbes themselves. However, because the genomes are generally millions of base pairs long, these 150 base pair reads only contain a very small subset of information in each genome. And that's where all of my computational biology and bioinformatics skills come into play. So from these millions of sequencing reads, we can start to ask questions about the microbial community that was in the original sample. And the first thing we can do is compare these 150 base pair reads. That's a database of all known microbes. And from this, we can answer the question of who's there and in what proportion. We can also take these 150 base pair sequencing reads, find overlaps between them, and use this information to construct larger microbial genomes. So for example, we can take all of the dark blue sequencing reads here and reconstruct the 3 million base pair genome of E. coli. And we can also look at microbial genes, either by assembling them from sequence or comparing against databases of known genes. And here, for example, we can identify 3 genes that we think are responsible for antibiotic resistance to 3 different antibiotics. And finally, we can attempt to predict functional pathways that are present in the genomes of the microbes that we study. This might be the ability to break down certain nutrients or produce certain metabolites. And this type of analysis moves the question from who's there, so what are the identities of the microbes, to really what are their functional capabilities and what are they doing. And this is what I really believe the future of microbiome research is going to be.
Ben, how long does it take to construct one of these, for example, the E. coli? And also to identify genes of interest. What's the time frame for you guys to be able to do that?
Once we have the data, it's pretty short. It's a very parallelizable process. So, a couple hours would be all it would take to run an analysis. Going back to the sort of, extracting the DNA and putting it on the sequencing machine, that's somewhere around maybe a week or so of time.
Okay, thanks.
And as a computational scientist, I'm pretty biased where I really like to study DNA. However, there are tons of other ways to measure microbes that are often used for the microbiome as well. First of which is bacterial isolation and culturing. So this is where we might be very interested in E. coli, for example, and we're going to take that sample and we're going to plate it out on a dish and we're going to enrich with specific media that E. coli likes to grow. And we will isolate very specific strains of E. coli from that mixed community. However, this is much more low throughput, where in the previous example, we get the DNA from all of the organisms present, but here we'd be only searching for a specific one. We can also use microscopy. We can actually look at what the bacteria are doing, and we can do that either in the sample, the raw sample itself, or after our culturing, isolation culturing. So here on the bottom left, I'm showing a phage, which is a virus that infects bacteria, and it's binding to this bacteria to infect it. And this is actually a photo I took on an electron microscope when I was in my undergrad. We can also study RNA and gene expression or proteins with different proteomics methods. So the central dogma of biology is that DNA makes RNA makes protein, and protein is the functional unit that is actually doing all the work inside the cell typically. So we can measure each of those things, but it's a little bit harder from a computational perspective, and it's harder to do in a mixed community that's the gut microbiome. We can use animal models like mice to test our different interventions. We can also, with different chemistry methods, we can measure the metabolites that the microbes are producing. So all of these kind of ways together, we can, depending on what your experiment design is, you can pick the best way to measure what you're interested in. So my contribution to the microbiome research that I focused on for my PhD is all about how humans acquire and transmit microbes. So I'm just going to go over a few of those stories now. I'm going to be fairly brief, but these are slides that I made for my defense talk. So there's maybe a lot more detail on them that I won't cover, but I'd be happy to go over them at length later. So the developing microbiome, as I talked about earlier, is best understood as a continuum from infants to adults. So as complexity increases with age, acquisition of new microbes falls. And we have some really strong evidence that mother to infant transmission of bacteria is responsible for infants acquiring their first microbes. And this is thought to be due to close contact between mothers and infants. And it's been shown that with mice models that don't have any bacteria in their gut, for example, they develop really aberrant immune systems that don't have the ability to fight off pathogens. So it's really important that infants get a healthy microbiome from their mother. And we have really strong evidence for this vertical transmission, from different genomic studies that have been published over the past few years. However, when we talk about adults, we don't really have any evidence for how adults either acquire new microbes into their fairly well-established microbiome community, or if adults are transmitting microbes between each other. There's a few studies that have looked at this, and I think they have pretty weak evidence so far. It may be true that spouses living together may be transmitting microbiome strains between each other, or siblings, or people living in the same household. But overall, the microbiome community of a healthy adult doesn't change much over time.
So quick question. Sorry, a quick question on the previous one. I think in the earlier you also mentioned that, for example, for adult, even they, for example, change their diet dramatically, the whole microbiome ecosystem still doesn't change that much in the long run. Is that because our internal system doesn't really change, even though we completely change our diet? Because you also mentioned that with different kind of, for example, nutritions, you can actually train to encourage some of the microbiome to change, right? To grow.
Right, and so I think that there are small changes that happen over time. And for example, if you eat a lot of seaweed, and a lot of Asian individuals have certain microbes that preferentially like nutrients that are found in seaweed. One of them is called porphyrin, and there are definitely interventions you can do that'll change small things. But I think the main conclusion, the way to think about this, is that an individual always looks more like themselves over time than anyone else, than they look like anyone else. So while you can change a few things of those 1,000 different species, most of them are going to be consistent over time.
Okay. Also, on the question, on the fact you said, if for example you're eating more seaweed, you're going to develop more bacteria or microbiomes actually going to benefit from the seaweed. Would it, on the other side, for example, it generates some sort of, I don't know, some sort of outcome or something, like actually stimulate the human to take more seaweed, for example, to benefit themselves? Is that such kind of loop?
Absolutely, and that's kind of what the whole gut-brain axis thing is about. When you kind of get it down into almost the philosophy of it, that are we ruling our microbes, or are our microbes ruling us?
Okay.
They're producing chemicals that are influencing the brain, that are maybe doing the things that are best for them in the long run. Those are some of the harder questions to answer.
Okay, cool.
So right, overall, we know mother to infant transmission happens frequently, but adults don't acquire new microbes either from each other or from the environment or from their diet too frequently. So I have two complementary stories where I studied transmission of a phage, which is a virus that infects bacteria from mothers to infants, and also transmission of bacteria between adults who are hospitalized for hematopoietic stem cell transplantation at Stanford Hospital. So the first of these studies was titled Acquisition, Transmission, and Strain Diversity of Human Gut Colonizing Crast-like Phages. So I'll get into what that all means in a second. This work was published in 2020. If you're interested, I can send you the citation later. But previous research had shown that mother to infant transmission of gut bacteria was common. And in a few different papers where I was able to get the data and actually analyze all of the sequencing that they did from these mother to infant pairs, they sampled, in this particular study, 44 mother and child pairs, and they collected stool samples from the mothers at 3 months before the day of birth and 3 months after birth. And they collected stool samples from the infants at 0, 15 days, 1 month, 2 months, 3 months, 3 months after birth. But the authors in these papers only looked at bacteria. So they showed that bacterial strains were commonly transmitted between mothers and infants, but it really kind of stopped short there. And our lab was very interested in this phage, so a virus that infects bacteria called crasphage. Crasphage was discovered via a metagenomic assembly process where I talked about you can assemble these larger genomes through this process called cross-assembly. So that's how it got the name crasphage, and the unfortunate capitalization in the center of the word. I don't know why they decided on that. But crasphage is really interesting to us because it was entirely undiscovered until around 2014. But it was very, very prevalent in healthy guts. So around 70% of people have this phage in their gut microbiome. But there's not really any known health consequences yet. So where people have looked for how having this phage in your gut is associated with inflammation or different dietary intolerances or things like that, there really haven't been any interesting associations. So maybe that CRAS phage is just kind of a marker for a healthy gut, and it's replicating on and killing bacteria without any real consequences for the human host. So when I started to look at CRAS phage in these mother and infant samples, first thing that we noticed was that CRAS phage was acquired at a young age. So in mothers from this one study, around 20% of mothers had the phage present at any one of their microbiome sample time points. Then we found infants started to acquire the phage in their gut microbiome at 1 month of age, and it increased all the way up through 3 months of age, which was the final time point from this study. In the second study I looked at, we found 25% of mothers had this CRAS phage present in the gut microbiome. And we found the first evidence in these infants at 4 months and then going up in proportion until 12 months. So the first conclusion from this research was that CRAS phage is acquired at a young age, but the proportion of infants that have the phage in their gut doesn't reach that of mothers by at least 1 year. So it may take longer. Then we started to ask the question if mothers and infants have identical CRAS phage. So we really want to find out if the entire genomes are the same between the mother and infant samples. And so we used this metagenomic assembly process I talked about earlier, where we took all of the sequencing reads that came from CRAS phage and combined them into the 100,000 base pair sequence that represents the CRAS phage genome. And we start to compare the sequences between mothers and infants. So we have a string of length 100,000 basically. And if we see greater than 99.99% matches between these two strings, we call these the same CRAS phage strain. And we find this as evidence for transmission because we would find the strain in the mother prior to birth and then in the infant after birth. And we think that direct transmission is responsible for giving this CRAS phage strain to the infant. And in cases where we found less than 99.99%, we thought we have a different strain here and transmission is not responsible. So in my research, I use a lot of this plotting method called a heat map. They're not too common, I think, for most people. It's outside of genomics, so I just like to explain them to everyone. But it's basically a pairwise way to compare several, or a way to show several pairwise comparisons. So we're just looking at a false color image of a pairwise matrix where we compared the genomes from each individual against all of the other genomes in the data set. And by definition, the diagonal of this represents 100% identity, so I grade that out. And so, for example, if you want to look at the value from comparing the genome from the mother from family 1 and the infant from family 2, you would find the row and column that represent those two samples and look at that box, and you would see that we have 100% identity here, and we would say that those are the same CRAS phage strain. But if we wanted to look at the mother from family 2 and infant from family 1, we would see a green square, and we'd see that's around 97% identity. 97% identity, and that's a different CRAS phage strain. So this is kind of one of the main results from the paper. It's a big heat map comparing all of the pairs of CRAS phage genomes from different samples. And so I put colors along the top of the heat map that indicate the different families that the samples came from, and the labels on the side are the actual individual that the sample came from. So we have 10 families with at least one mother and infant sample, and 8 out of these 10 families cluster together. So this clustering is just done based on the pairwise comparison of those CRAS phage genomes. And in 4 families, we find completely identical CRAS phage genomes in both the mother and infant. And that suggests to us that we have direct transmission is responsible for this phage. So some conclusions from this work. We found that CRAS phage can be acquired starting at 1 month old, but it may take longer than 1 year to be fully prevalent in infant populations. We also found that infants born via C-section have reduced prevalence of CRAS phage, which is another interesting finding. We found that CRAS phage is likely to be directly transmitted from mothers to infants. And this kind of raises the questions that CRAS phage may be a marker for the maternal lineage over time, much like mitochondrial DNA is. So you can think if you got your CRAS phage from your mother and your mother got it from your grandmother, it may be sort of this marker that's passed down over time through many generations. And then the last point about strain diversity in the crassophages, something that I didn't talk about here, so you don't have to worry about it. So wrapping up the mother and infant work, the next project I worked on and published recently was titled Rare Transmission of Commensal and Pathogenic Bacteria in the Gut Microbiome of Hospitalized Adults. So here we're no longer talking about infants, but we're going to be talking about adults, where we think that transmission is fairly rare and the adult microbiome community is well established. But at Stanford in the bot lab, our interests extend beyond just the general microbiome and into the clinic. And we studied patients undergoing hematopoietic stem cell transplantation, or HCT. And this is a process that typically involves transplantation of donor bone marrow or potentially curative therapy for hematologic malignancies like leukemia and lymphoma. But the patients undergoing this process are hospitalized for a very long period of time, from weeks to months. And there's many complications in the process, including graft versus host disease and infection. And infections are responsible for 22% of the deaths after HCT. And that's more than any other factor other than the cancer relapse. So we were really interested if we could potentially be preventing infections by studying the patient's microbiome, where many of these actually infectious organisms reside. So as for an example of why the microbiome matters in the clinic, I really like to use this example from a patient that we had at Stanford. So here I've collected 5 stool samples from this patient over a period of 2 months roughly, and the day is indicated on the top. In each sample, the relative height of each bar of color represents the proportion of that different species in the microbiome sample, and the color represents the species. So what I hope you can appreciate here is that this patient came into the hospital with a healthy, diverse microbiome, underwent transplant on day 2, that their first 2 samples have a lot of different colors. However, in the middle 2 samples here, this patient experienced intestinal domination, the pathogenic E. coli strain. So, you can see the light purple here is almost the entire microbiome, and that is an E. coli that's antibiotic resistant and producing chemicals that are probably not very good for this individual's gut. This patient then experienced an E. coli bloodstream infection, and thanks to work by Fiona Tamburini in our lab, we actually compared the genome sequences of the bloodstream infection and the E. coli that was in the patient's gut prior to the bloodstream infection. She showed that they were totally identical, which means that the E. coli infection probably came from the patient's own gut, and it probably came across the damaged intestinal barrier. As you saw in the picture I showed at the beginning, the microbes are really, really close to the intestinal cells, and if that barrier is damaged from chemotherapy or radiation or other things that the patients are experiencing, you can actually get translocation of those bacteria across that barrier, and it can cause infections. But luckily, this patient recovered after antibiotic treatment for their infection, and later their microbiome recovers diversity as well. As you can see, there are nice, healthy colors on the end sample as well.
So how much of that, it's interesting, but I'm curious, during those days, the 28 to 34 days, what kind of medication or what kind of treatment did the patient go through, and how much of that have an effect on the ecosystem of the microbiome in order to have it, like let's say lack of nutrients, right? So then that may be also the case as well, I would assume.
Yeah, definitely. A lot of these patients have fairly bad graft versus host disease, which manifests on a lot of so graft versus host disease means the transplanted immune system that the patient has received is attacking the host immune system and attacking the host cells. And this often manifests in tissues that reproduce frequently, like the lining of the mouth. So this makes many patients unable to eat any solid food or any food at all. And many of them are on just IV nutrition. And so this is one of the major things that can contribute to a lack of diversity in these patients. Also, they have prophylactic antibiotics that they take when they come into the hospital. The radiation treatment does a lot of bad things for them. It's really a combined barrage.
So on the last column, right, even though this patient recovered, right, with a very diversified microbiome, but you can see actually it's very different from the before this patient did this procedure, right, from the first column. There is a big different color, like shifted. like
right.
dark brown and red.
The dark brown is new. The proportions are different. And unfortunately, what this chart doesn't show is, the number of lowly abundant things that are kind of shoved into the gray other column. Those are very different. And then when you actually look at the genomes that are in the early and late samples, they're very different. So it's not, even though the species identity is the same, it's not the same microbe, it's not the same population. And so it's not like something persisted over time. It is really new colonizers that have come in.
Do you think after maybe, let's say after 2 years, 5 years, eventually the balance will shift back to the original, more like the original, or completely change forever?
Yeah, that's a good question. And we don't have any data beyond like 90 days in these patients. I think most of them are pretty different for a long time because, what I was talking about, diet doesn't change microbiome too much. No one in their normal course of life is experiencing things like this, where you're taking these horrible antibiotics and chemotherapy and killing off all the things. So I think a lot of these patients are going to have something very different. They also get a new immune system, right? The stem cell transplant gives them a new immune system. So the way that the microbiome tolerates, or the immune system tolerates certain microbes is going to change. So if we think about ways that we may be able to modify the microbiome in the clinic, we'd really like to prevent this patient from either acquiring this E. coli pathogenic strain or being dominated by it and it leading to the translocation into the bloodstream and causing the infection. So we're left with a few questions about why this E. coli was here and where it came from initially. So a few options for this E. coli. It could have been present in the gut of this patient when they came into the hospital. That was not the case in this patient. We looked at the day 0 sample and we can't find evidence for it. This patient could have acquired the E. coli from the hospital environment. So it's known that pathogenic bacteria can live on surfaces and in sinks and different places throughout the hospital. And perhaps the most interesting possibility for this E. coli strain was that it could have come via horizontal transmission from another patient. So patients, as they recover from transplant, often have roommates at the hospital here. And so it's possible that patients who are living in the same room and using the same bathroom, as their microbiome is being perturbed and recovering, they could be picking up new strains from the person that they're in the same room with. So we wanted to study this at Stanford, and we began by examining the electronic health records of all patients who were in the hospital for a transplant over a period of 3 years. So on the left here is a blueprint of the HCT ward at Stanford. There's 18 rooms, but 4 of these rooms are double occupancy. So there are 2 patients in the room. Those rooms are underlined here. And so when I looked at the electronic health record data from these patients, and I grabbed all of the time stamps and the rooms that they were in, we can draw a network of all patients who are roommates for at least 1 day. So shown here on the right is that network. Each patient is a node, and I've drawn an edge between patients if there's greater than 24 hours of overlap between them. And the patients are colored from the type of data that we have from them. So we have stool samples or we have sequenced stool samples. And what really surprised us was that 73% of patients had a roommate at least once as they recovered from transplant. And 77% of patients were in the largest component on the top of the graph here. And so this is potentially a network along which these infectious agents could be spreading over a period of 3 years. So what it's like working in a poop lab that studies the microbiome, we've been collecting weekly samples from patients in the hospital going through transplant, going back to 2015 or so. So we have over 2,000 vials of stool from over 900 patients in our freezer, and that number was accurate as of about a year ago, so that's continuing to grow. And so roughly 300 of these samples had been previously sequenced. On the right here is my advisor, Ami Bhatt, and she's looking at some stool samples in the freezer. To add to this 300 stool samples, I picked 96 new samples to maximize the coverage of roommate overlaps, as we saw on the graph on the previous slide. And I performed this technique called linked read metagenomic sequencing. And this is a technology from 10x Genomics. And it basically gives us 10 times better assembled genomes. So the very long E. coli genomes that we get out of our microbiome data is roughly 10, each individual one is roughly 10 times better. And when I look across all samples, all 400 samples that we analyzed, we found roughly 1,500 unique species level genomes that I was able to assemble from the large collection that we had. So that's roughly 1,500 different species that were present across all of the 400 samples in our collection. I'm just going to skip this because that's too many details. But in short, we were looking for transmission, and we mapped sequencing leads from 400 samples against the 1,500 unique species level genomes. And we compared the strain populations between all pairs of patients. In short, we found a rare transmission of both pathogenic and commensal bacteria between patients who were roommates as they recovered from transplant. As we were interested in pathogens in the first place, with the interest in protecting patients from these infections, the first microbe that we found to be transmitted between patients was Enterococcus faecium. This is a gram-positive coccus shown here on the left. It's a facultative anaerobe, so it can't really live. It can actually tolerate oxygen and can potentially live outside the host for certain periods of time. This microbe has frequent resistance to the antibiotic vancomycin, and it's actually the most common pathogen to dominate the microbiome of our patients in the hospital, and it's the third most common cause of infection in our patients. And when I compared Enterococcus faecium strains between patients who are roommates, I found a few interesting cases of transmission, and I'm just going to show one of them here. So on the top here, I'm showing the microbiome composition of one patient that we collected stool samples from on day 0 and 14. On the bottom, we collected a single sample from this patient on day 14. And these patients were 10 days, they spent 10 days in the same room and 14 days together total in the hospital for any period of time. The top patient had a microbiome almost entirely dominated by this Enterococcus faecium strain, but the bottom patient only had this Enterococcus faecium at 0.4% relative abundance. So actually, when we compare the genomes from the top and the bottom patient, we're able to assemble 2.3 million base pairs of Enterococcus faecium sequence in both, and we actually found no differences between the two genomes. So that's no SNPs, no genome changes between the top and the bottom patient. We don't have really good time course sampling here, so we can't say that the strain definitely came from the patient on the top. But to me, that this sort of direction seems to be the most likely. We also found a few interesting cases where we saw transmission of commensal microbes as well, and this was contrary to our expectations. We thought we'd just be seeing pathogens. The first was Hungatella hathawayii, which is this interesting rod-shaped bacterium, but it can't usually live outside the host. It can't usually tolerate oxygen, except when it goes into a spore-forming state. And from this, it can actually form spores and persist for long periods of time in an oxygen environment. So in this example, we have several samples from the patient on the bottom and only 2 samples from the patient on the top. These patients spent 34 days together in the hospital, but only a single day as roommates. And when we compare the Hungatella hathawayii strains between our patients here, we saw almost totally identical strains between the top and bottom, indicating that we think there was a transmission event between these patients during the period of time in which they were roommates. That's up here on the top left. So in this case, we have really good dense time course sampling. We don't see this orange strain in the microbiome of the patient prior to this transmission event, and we really think that direct transmission between patients who are roommates is responsible here. So interestingly, this patient on the bottom, sorry, did you have a question?
Yeah, I have a quick question. Since this could be a pass, then does that mean, for example, the same family members could also have this kind of transmission often?
Yes, I think that definitely happens between family members. And I think if this patient was at home with their spouse, rather than picking up strains from their roommate in the hospital, they'd be picking up potentially like healthier strains that were around the family. So that's one interesting thing we could start to think about. But really why this transmission happened, I think, is because this patient on the bottom had a bloodstream infection with the Streptococcus species. And then for this infection, they took extensive antibiotics. And this sample on day 34 was very simple compared to their previous samples. So it was killing off the natural colonizers that let this new transmitted microbe come into their gut.
What's the route of transmission?
Say again?
How does it get transmitted? What's the route? I mean, what's the different methods of?
So this particular Hungatella hathawayii microbe is interesting because, like I said, it forms spores. So I think, the route is the bathroom that is shared between these patients, whether that's, microbes kind of as you flush the toilet, microbes kind of spraying out as it happens all the time and coating a surface that a patient then picks up later, or some other kind of, surface, fecal to oral surface mediated transmission, I think is what's responsible here.
Okay, so that's interesting. So, okay.
Yeah, I think one of the kind of conclusions that I've come to as I've studied the microbiome is that, there are bacteria everywhere, and we are very politely and happily ignorant of where they are and how much they're spreading between surfaces.
Yeah, but essentially, in order for us to go into our body, has to go through, is it oral, right? So let's say if you touch a surface of anything, the bacteria essentially, when you eat something, that's the it has to be transmitted via oral ingestions or whatever the case is, right? But never through, like, say, your skin or
right. To get into the gut and reside and continue to grow, it has to come fecal to oral to the recipient's gut. But like in contrast to infections that may be spreading from the skin.
All right, cool.
Finally, one of the interesting things we also saw in this data was that we found totally identical strains of this species called Lactobacillus rhamnosus. And we found identical strains in 8 patients over 3 years, but none of these patients were roommates, so we didn't really have any evidence for transmission here. But what was interesting was that Lactobacillus rhamnosus is a common commercial probiotic. So we think that all of these patients were taking potentially a probiotic as they recovered from transplant. That's interesting because their doctors tell them specifically to not take probiotics because it is potentially an issue if you're introducing new bacteria when you don't really have an immune system as you recover from transplant. But the patients as they recover also aren't allowed to eat high bacterial foods like active culture yogurt or soft cheeses and things. So it's just some interesting conclusions that we found in our data of how the patients may be violating their doctor's orders. So some final conclusions, we found that these patients frequently harbor pathogenic organisms in their gut microbiome. These pathogens may be rarely shared between patients. We found evidence for Enterococcus faecium transmission, but not E. coli. We found commensal microbiome transmission under specific conditions that included antibiotic exposure after infection and patients who are roommates with a shared bathroom. And finally, I'll pose the question that having a roommate may not necessarily be a bad thing. So as you recover diversity after your antibiotic exposure and such, it may be actually helpful to have these sort of microbes around. But in this case, it may be best that you have your friends and your visitors and your spouse in your room and not your also very sick roommate as they're recovering from transplant. And they're actually starting to do stem cell transplants at home now, or at least the recovery process at home with some pretty strict watches on immune system function and all that kind of stuff. So it may be best for the patient to actually be in a friendly setting as they recover. So the final conclusions from this work, we found that mother to infant transmission of phages along with bacteria was also pretty frequent. And we also found that patients who were hospitalized for stem cell transplant could occasionally transmit bacterial strains between each other. And this is actually the first genomically characterized time course resolved example of microbiome transmission between adults. So as I said earlier, there's some weak evidence that this happens, but I was really the first one to characterize that adults were able to do this and not just mothers to infants. I'm just going to skip this section. So kind of moving on into where I see the microbiome fitting into biotech and personalized medicine. I think there's a lot of potential for microbiome therapeutics, and that's not just using bacteria as a therapeutic, but also shifting the microbiome to a way that can help a patient perhaps recover from some disease better, or shift the microbes in a way that leads to less inflammation. So for public companies, there's a few out there. One I've followed closely for a while is called Series Therapeutics, and they're in in Cambridge, I believe, in Boston. They have a few different treatments out. One of them is a treatment for Clostridium difficile, and that is it's actually a spore formation. So it's microbes that are kind of dried down into spore forms, and they're given to the patient if they have this recurrent Clostridium difficile infection. And this is actually passed a phase 3 trial maybe about a year ago, showing that it had significant effect for resolving Clostridium difficile infection. They have a few other programs out there for ulcerative colitis. Infection and graft versus host disease, as I talked about, is a problem for patients recovering from stem cell transplant. Unfortunately, their ulcerative colitis drugs have not been performing very, very well. So I think that they have a few challenges to come over. But this is the main company doing live or spore form the actual therapeutics with microbes. Finch Therapeutics is another one in Boston as well. They're also working on recurrent C. diff. I think most microbiome companies are going after C. diff as the first thing that they're going to study. But then other diseases of the gut as well, including ulcerative colitis and Crohn's disease. Again, using either rationally designed or whole microbial consortium that they're going to be giving to a patient. They're going after autism spectrum disorder and chronic hepatitis as well, in sort of non-gut related diseases. Final microbiome kind of themed public company is Kaleido Therapeutics. They're a little bit different, where they're working on a technology called microbiome metabolic therapies, or MMTs. And so this is kind of like the prebiotic concept that I talked about earlier. So Kaleido is focused on medicines that they can give to people that'll promote the growth of healthy gut microbes rather than actually giving the microbes themselves. So these might be nutrients or signaling molecules that help certain bacteria grow stronger. I think they have some interesting potential, but they're all still in preclinical studies. And finally, I think the most interesting and most likely to be successful biotech company is called Novum. This is a company that started out of the Sonnenberg lab here at Stanford. They raised a Series A, I think $33 million Series A last year. So they're doing very well. They're starting their first in-human trials, I think, now. They have this concept called genetically engineered microbial medicines, or GEMS. And this is really brilliant. And they don't have a great diagram of the way this works on their website. I wish they did, but the GEM concept really solves the colonization and engraftment issue that I talked about with probiotics, where the bacterial strains don't persist long term. So what they're able to do is take a bacteria and they're able to engineer its genome so that it is entirely dependent on an oral polysaccharide pill that they can give someone. So they give this pill to the patients, the microbe, because it is the only, it's like a unique nutrient that only this microbe can use. That microbe grows throughout the gut really successfully. And it also gives you a kill switch, because if you need to stop this microbe from being in the gut at any point in time, you can just stop giving this oral pill and the microbe will die off. So you have both the ability to make sure it engrafts and the ability to kill it if it becomes a problem. And they've also engineered these microbes to produce a therapeutic enzyme. So they're going after hyperoxaluria, which is a disease caused by high oxalate in the urine and leads to kidney stones and kidney dysfunction. So they've also engineered these microbes to have an enzyme that degrades oxalate. So they really have like the key to rationally designed microbial medicines. And I'm really curious where they're going to go.
Interesting. So for the previous for these couple of companies, I mean, the way it works, I just maybe you can a little bit very, very high level, is all very similar to the last one. I think I understand what you said. They basically encourage some of the microbiome grow, right? And then to benefit, so the human can benefit from it. How about the first couple? Are they also with the same way, the way it works?
Sorry, I should have explained that a bit better. For Series Therapeutics, for example, that they're actually doing live bacterial therapeutics. So they have either single strains or communities that they're going to give people that have this disease. That in the case of Clostridium difficile, it's you have a persistent and antibiotic resistant sort of community of this C. diff in your gut that can't be displaced by antibiotics, and it's causing you a lot of inflammation and diarrhea. And so they're going to give you this healthy community that's going to displace the harmful C. diff communities in there.
Let's see. Great.
Finally, there's a few companies focusing on personalized medicine in the microbiome. There's more than a few. I think there's a lot of hype around the microbiome. You'll see a lot of claims being made about being able to predict your favorite type of foods or favorite type of wines or things based on your microbiome composition alone. And I think some of those are scams, but there's a few gems in the rough here. I think one of the most interesting things I've seen out of this is out of the Aaron Segal's lab in the Weizmann Institute in Israel. And so here they were able to predict your response to, or your glucose response to certain meals entirely based on the microbiome content of individuals. So they profiled people over time where they wore a continuous glucose monitor, and they record the diets they ate and also their microbial features. And they're actually able to design specific diets that are supposed to normalize your blood glucose levels over time. And the only input to this machine learning model after they trained it would be our microbiome composition. So I think this was some really, really cool research, but this was 2015. So a lot of things have come along since then. And I think they're building a company off of this, but I haven't seen anything since. So if anyone knows what has happened with this personalized medicine research, I would love to find out.
So they founded a company called Day2.
Okay.
And they try to screen the microbiome for the healthy or unhealthy people, and give them a preference, give them like what you need to get more from the diet, give them very, very general guidelines, and not precise for the glycemic response, not as precise as the original research. Yeah.
It's called Day2?
Yes, Day2.
Okay, great. I will look into that. Thank you. One other interesting company I've found is called Zoe. They're doing microbiome sort of personalized diets. So the goal of this company is to understand how your body responds to food better. So whether that's your energy levels, your sort of weight gain or loss, your glycemic responses, etc. So they do at-home tests, including a blood and a fecal sample, and through some fancy machine learning algorithms, they will tell you what your best supposed diets are. This is just one example. I think there are a few companies out there that do something similar to this, none of which I've actually tried. So I'd be interested to see how well they work. That's all I prepared for today. So I thank you all for attending. Here's two pictures of my lab with me and them. One from, I think, 2018, and then one from about a month ago. So we've certainly grown in size, and we've seen a few people graduate and defend since then. But thank you for coming to my talk and listening to the microbiome. And I'm happy to take any questions about microbiome, biotech, etc.
Great. This is great. Questions? Maybe I'll jump on first. So do you see the specific, like personal medicine wise, right? Do you see any new directions, potential use cases for the personal medication for using microbiome?
Yeah, I mean, there is kind of like a holy grail here, right? Which is, we take the sample, we tell you what you can do to make it better, to normalize your blood glucose, to help with your inflammatory bowel disease and things like that. I think there's going to be successes in the field, but I think it's really hard. Part of which due to the fact that I think a lot of people aren't measuring the right things about the microbiome. So in a lot of cases, we're only looking at what species people have and what proportions of these species people have. And we're not actually looking at the functional capabilities of these microbes, for example. So there's definitely like innovations that need to happen on the discovery side, and also innovations that need to happen on the ways that we can modify the microbiome. Like even if we know what you should have, it's hard to get it to change that way. Like I talked about, probiotics don't really engraft long term. So I think there was like a lot of hype about the microbiome in 2015 through like 2019, and I think now it's kind of the hard research has to start. And in order for some company to be successful, they have to tackle a lot of these issues.
That's great.
Is there any, do you know if there's any research or any study being done to evaluate the kind of just the drug interaction with the microbiome environment, and how's that going to be able to just think long term in the future, how that even to have if you can think about increasing the efficacy of let's say any of the drugs out there potentially, do you think microbiome, understand more about the drug interaction with microbiome would help or would be an important part? And any companies are working on.
Yeah, I think that is going to be a big thing. There have been papers published showing that the microbiome can degrade certain drugs that you take, reducing your concentration. As for any companies out there, I don't know any yet. I would love to work on a company that could, for example, predict your response to a certain drug in the clinical trial based on your microbiome, or at least look and see that, oh, we know that this drug can be metabolized by certain bacteria, and as you take this pill, the concentration is going to go down because certain bacteria are eating it. But I'm not aware of anyone doing that yet.
Okay, well, talk to me in a year or 2 years. Maybe we can do something.
Yeah, I think there's potential there for sure.